AACR 21, several personalized medicine and precision oncology studies were presented. We picked three important studies. The first one, the Libretto 001 trial of selpercatinib in RET fusion positive all cancers. The second one was the phase 2 Phoenix CCA2 trial with futibatinib. And the third trial was the ROAD study, which is a rare oncology agnostic research with dabrafenib and trametinib in high-grade and low-grade gliomas. During AACR 2021, I had the pleasure and the opportunity to present the initial results of the efficacy and safety of selpercatinib, a highly selective RET inhibitor in patients with RET fusion positive cancers other than lung or thyroid cancer as a part of the Libretto 001 clinical trial. The RET proto-oncogene is generally activated by one of two mechanisms, RET fusions or mutations. RET fusions predominantly occur in 2% of lung cancers, 10 to 20% of all thyroid cancers, and in a very low frequency in an increasing number of diverse cancers, including difficult to treat pancreatic cancer, colorectal cancer, and rare cancers like salivary gland cancer. The therapeutic relevance of RET fusions occurring outside of lung and thyroid cancer has not been well established. Selpercatinib has received approval in multiple countries, including the United States and Europe, for the treatment of RET fusion positive lung as well as RET-driven thyroid cancer. The Libretto 001 clinical trial enrolled a total of 441 patients with RET fusion positive cancers that include non-small cell lung cancer, RET positive thyroid cancer, in addition to 38 patients who had a RET fusion positive cancer beyond lung cancer and thyroid cancer. What we presented at AACR was the non-lung and non-thyroid cancer RET fusion positive cancer patients. 62% of the patients had treatment refractory GI cancers with very limited therapy options. What we found in the study, RET fusions in tumors other than non-small cell lung cancer and thyroid cancer are uncommon but potentially actionable. Selpercatinib demonstrates promising activity across a wide variety of non-lung and non-thyroid RET fusion advanced solid tumors like breast cancer, like RET fusion positive pancreatic cancer, and rare diseases like metastatic xanthogranuloma, including treatment refractory GI malignancies. The objective response rate by investigators' assessment in this heavily pretreated population was 47% and included complete responses. Efficacy was observed across cancer types and across a spectrum of RET fusion partners. Overall, selpercatinib was very well tolerated with a safety profile consistent with what has been previously reported, with no new safety signals identified. What does these analysis tell us? This analysis reiterates the importance of broad-based genomic profiling to identify actionable oncogenic drivers, including RET fusions. At AACR 2021, one of my colleagues from Boston, Dr. Lipika Goyal, had the pleasure to present the results from the phase 2 Phoenix CCA2 trial, which is a single-arm, multi-center phase 2 study evaluating futibatinib in patients with intrahepatic cholangiocarcinoma harboring the FGFR2 gene arrangements, including gene fusions, who have failed at least one line of therapy. What did the study show? In the Phoenix CCA2 trial, 103 patients with locally advanced or metastatic unresectable intrahepatic cholangiocarcinoma patients harboring FGFR2 specific gene arrangements, including fusions, who had received at least one or more lines of systemic therapy, received this FGFR inhibitor called futibatinib, 20 milligrams once daily until disease progression or unacceptable toxicity. The study met its primary endpoint of a greater than 20% objective response rate as assessed by independent central review of an objective response rate of 41.7%. The secondary endpoints of duration of response and disease control rate were also reported as a part of the study. Responses were durable with a median duration of response of 9.7 months and 72% of responses greater than 6 months and a disease control rate of 82.5%. Interestingly, the median progression-free survival was 9 months and the median overall survival was 21.7 months with 72% of patients alive at 12 months. Common treatment-related adverse events were hyperphosphatemia, which is an FGFR class effect in almost 85% of patients, alopecia in one-third of patients, and dry mouth in one-third of patients. The most frequent grade 3 treatment-related adverse events was hyperphosphatemia, again, which is a class effect of FGFR inhibitors, which result in patients with adequate management. One grade 4 treatment-related AE of increased ALT, which is a liver function test, was reported and there was no treatment-related death. The results of the Phoenix CCA2 trial are significant for patients living with this challenging disease with refractory intrahepatic cholangiocarcinoma with FGFR2 gene fusions or other gene rearrangements, as futibatinib showed a meaningful objective response rate of 41.7% and a good durability of response. These results represent another example of the promise of precision medicine in cholangiocarcinoma, which has become a poster child of precision oncology. Interestingly, last year in April 17, 2020, the Food and Drug Administration given accelerated approval to pemigatinib for the treatment of adults with previously treated unresectable cholangiocarcinoma. In addition to pemigatinib, futibatinib could be a potential future options for patients with refractory cholangiocarcinoma if approved. At AACR 2021, I had the pleasure and opportunity to present the results of the ROAR basket study. What is ROAR? Rare Oncology Agnostic Research. Dabrafenib plus trametinib in BRAF V600E mutant high-grade glioma and low-grade glioma. Gliomas are the most common primary tumors of the brain. There are approximately 6 cases per 100,000 people each year in the United States. As we all know, more effective treatments are needed to improve the outcomes in both high-grade glioma and low-grade gliomas. BRAF V600E mutations specifically have been identified in 5 to 15% of low-grade glioma that include pleomorphic xanthogranuloma, ganglioglioma, and pilocytic astrocytoma, and less frequently in high-grade glioma, including approximately 3% of difficult-to-treat glioblastoma. Dual blockade of MAP kinase pathway using the BRAF inhibitor dabrafenib and the MEK inhibitor trametinib is standard of care in BRAF V600 mutant melanoma, non-small cell lung cancer, and a rare disease of the thyroid gland called anaplastic thyroid cancer. This is a non-randomized, open-label, phase 2 basket study of dabrafenib and trametinib in patients with BRAF V600E mutant positive rare cancers. Here, in this trial, we report the results of dabrafenib and trametinib in high-grade and low-grade glioma. Meaningful efficacy was seen in patients with glioblastoma with an objective response rate of 32%, a median overall survival of 13.7 months with CI of 8.4 to 25.6 months. The safety profile was consistent with the known safety profile for other approved indications of dabrafenib and trametinib. Interestingly, no new safety signals were observed. A comprehensive next-gen sequencing showed a heterogeneous landscape in glioblastoma and low-grade and high-grade gliomas and a low tumor mutational burden. What is the summary? BRAF V600E is an actionable driver mutation and should be considered for routine testing in glioma patients that include both low-grade gliomas and high-grade gliomas.